responds to it. Those are the two major technological separations, because you can imagine that making a protein in a manufacturing process will be quite different from making a genetic code in a manufacturing process. So those are the categories that we should think about. In the first category, make the protein outside and then inject it, we have the inactivated virus. That's the simplest way of making the protein outside. You just make the whole damn virus, which of course includes the protein that you're interested in, and the protein that we are interested in we'll come to in a minute. But you therefore just take the virus and you inject the virus, except that you don't want the virus to actually cause the disease. So you inactivate the virus so that it is no longer infectious. You do it by chemical inactivation by and large, by a variety of chemical interventions, and you can inject that. And there's 3-4 different COVID-19 vaccines with inactivated virus as the approach in advanced phase 3 clinical trials, including the ICMR Bharat Biotech Covaxin that there is so much of conversation about. But there are already, I think, 2 or maybe 3 vaccines from China that are in phase 3 clinical trials. One of the odd vaccines, maybe from Kazakhstan or somewhere, is also in clinical trials. And certainly one of the Chinese vaccines has already shown fairly good evidence for protection, early evidence for protection. So you can inject the whole virus, or you can actually take the genetic code of the particular protein that you are interested in. And since we are interested in stopping the virus from getting in, the target of the immune response that we want is that particular protein of the virus that allows the virus to bind, to stick to cells. So that's the spike protein, and therefore the spike protein is what all 200 vaccine making efforts are using as the target. So you can take the genetic code for the spike protein, but instead of injecting the genetic code, you can use so-called genetic engineering industrial technology and get bacteria or yeast to make the protein in industrial scale fermentation methods, purify the protein and inject that. Those are what are called protein subunit vaccines or virus-like particle vaccines and so on and so forth. In the inject the genetic code technologies, either you can put the genetic code in the backbone of a virus, because what you need to inject along with the genetic code is an instruction for the body cells to make a lot of the protein. So the simplest way of doing that is to use the instruction, the genetic instruction from some other virus. So that's why adenoviral vector-based vaccines, such as the Oxford AstraZeneca Serum Institute vaccine or the Gamaleya Institute vaccine, or a whole bunch of other vaccines that are in development, use a whole adenoviral backbone, but the adenovirus is defective, so it's not really a virus infection. It's just that the backbone is being used to give the instruction to make a lot of protein. Or you can use a very pure instruction hooked to the genetic code, and that's what's being referred to as the DNA vaccine, which is what Zydus Cadila in Ahmedabad is in phase 3 clinical trials for. Or keeping in mind the old classical high school biology that DNA is made into RNA and RNA is made into protein. So far, we've been talking about injecting DNA-based genetic code, but we can also make the RNA of the genetic code and directly inject the RNA. And that's what the BioNTech Pfizer vaccine or the NIH Moderna vaccine, which are called RNA vaccines, is doing, injecting the RNA. But the end result of all of this is introducing viral protein into the body for the immune response to target and to be activated. In all of these, remember the second part. We need to cause a little bit of damage so that the immune response wakes up and begins to act efficiently and with high magnitude of response. And all sorts of so-called adjuvants, additives that cause just a little bit of inflammation, will be added. And that's been one of the controversies regarding the Bharat Biotech Covaxin, about what adjuvant has been added and so on and so forth. But that's the gamut of vaccine technologies. In technological terms, there are very interesting pros and cons. But maybe we can take another question and then talk about that.
Yes, that is what I was going to come to. We have all these different types, but really, so for instance, we have this mRNA-based vaccine, which is, it's for the first time that this type of technology has been approved against any disease. So really, what makes this technology better? Why was it, what were the challenges of using it? Why was it not approved before? And what are the disadvantages it has? And can you tell us about that? And also a general comparison of all of these different types of vaccines.
Okay, so let's talk about the inactivated virus vaccine as the simplest technology, the oldest technology, the simplest technology. The obvious limitation is you're growing a disease-causing virus. You're growing it at industrial scale. A great deal of safety protocols need to be put in place for containment. That becomes a major industrial limitation on making these vaccines. Now, keep in mind that SARS-CoV-2, the COVID-19 virus, is not actually hugely dangerous at the individual level in the same way that, say, Ebola virus is dangerous. Out of 100 people catching SARS-CoV-2 infection, maybe out of 400 people catching it, maybe one will die. Out of 100 people catching Ebola infection, more than 30 are likely to die. So we are talking about dramatic differences. But even so, a disease-causing virus being grown at the industrial scale needs a lot of containment. So that's a limitation, if you like, of the technology. Secondly, live viruses are grown in tissue culture of human cells. That, again, is a technology that's not easy to scale up to massive amounts. So there is a scale limitation with an activated virus vaccine. And we will see this playing out in the Indian market today, based on the straightforward calculation about comparing the number of doses that the Serum Institute of India can provide of the adenovirus-based vaccine called Covishield versus how much Bharat Biotech can provide of the inactivated virus vaccine called Covaxin. So that's one limitation. The second way is to make proteins in a test tube and then inject them. Now, the trouble with making proteins in a test tube is it doesn't have any of these limitations of the inactivated virus, but it acquires a different limitation. And that is, proteins are huge molecules. In terms of, in a chemist-eye perspective, proteins are enormous molecules. And proteins need to fold in appropriate shapes in order to look like themselves. So the immune system needs to see a properly folded protein if it's going to make an immune response that will also recognize the same protein on the virus. And this folding of the protein is industrially a little tricky. So optimizing protein folding as an industrial step of the technology is not as simple and straightforward as it sounds. And that becomes a limitation. And in fact, that and its variations have been the limitations for example, Novavax, which is making this kind of a protein vaccine, having gotten a little bit delayed in its clinical trials. They needed to figure out the technology, the scale-up technology. At the laboratory, it's not difficult, but the scale-up technologies tend to be a little tricky. So those are the kinds of problems that those technologies have. On the other hand, the genetic code doesn't actually, because it's being read as a sequence, it doesn't need to be folded into anything. We can synthesize huge quantities of the genetic code, and it's therefore easy and simple. And this is why the mRNA-based, the adenoviral vector-based, and the DNA-based vaccine technologies have come into play much more rapidly, because they don't need to worry about the folding problem, as protein chemists call it, at the industrial scale. So then the obvious question is, then why haven't we seen RNA and DNA and adenovirus vector-based technologies deployed? Why are they the first time they are being deployed? And the short answer is that we really came to them being industrially viable only over the past 15 or 20 years. And over the past 15 or 20 years, we haven't really made and deployed a lot of vaccines the world over. So let me give you, therefore, an example that connects science and technology to industrial manufacture and politics. So over the past 15 or 20 years, let's remind ourselves of the viral infections that have caused major concern. And all of us will remember this. Even you in your youth will remember some of this. We've had Zika virus. We've had the original SARS virus. We have had yet another cousin coronavirus, that is the MERS virus. We've had Ebola. And interestingly, over the past 20 years, against all of these incipient dangers, we've had RNA vaccines developed, we've had DNA vaccines developed, we've had adenovirus vaccines developed, right? So the obvious question is, then why didn't we see them deployed for all those diseases? And the answer to that is that vaccine development is a quintessentially capitalist process. And as a consequence, the question was not for deployment of these vaccines against those diseases. The question was not, is there a need out there? The question was, is there a sufficiently large need out there to provide a sufficiently large profit to justify the investment in deployment? And the answer so far has been no. For the first time in COVID-19, the answer is yes. And that's why, in a science tech political landscape, we are seeing the first time deployment.
Right. Thank you, Dr. Satyajit, for joining us today for this very enlightening and interesting discussion. That's all the time we have for today. Keep watching this clip.